So I should mention, these are things you're not necessarily going to know, but genome evolution is quite complicated. I think most people think about genome evolution as just being mutation, right? You change one nucleotide to another, but there's all sorts of things that occur in the genome. You can have deletion events, that is to say some parts of the genome can be deleted, or you have insertion events where you have new genetic material being inserted into it. You can have rearrangements, right? The genome is filled chock full of what we call parasitic genetic elements, that is to say genetic bits of your genome that self-replicate, and a lot of the genome, most of your genome is actually parasitic. It's not functional, which is crazy to think about. But one thing that can happen, for a number of reasons, is you can have a messenger RNA, which is reverse transcribed, that is, goes from messenger RNA to DNA and reinserted into the genome. What that means is you often have a gene or a DNA sequence that looks the same as a gene, but doesn't have any of the regulatory machinery that allows it to be expressed. So it's called a pseudogene. You have the functional gene somewhere in the genome, and then you have this copy of this gene that just has the bit of the coding gene, but none of the machinery that allows it to be transcribed and expressed. Okay? So that's called a pseudogene. And what happens with these pseudogene is, well, because they're not functional, natural selection doesn't maintain them, and they just basically they over time mutation sort of erases the signal. Over time, you can't even, if given enough time, you can't even recognize the pseudogene as being related to the functional gene that it came from. Right? They erode over time. But they're chance events. When you have a pseudogene being created, it's a chance event. Right? So here's some examples of pseudogenes. Here's you can see that the alpha enolase pseudo-Cy1. Psi means it's a pseudogene, and it's the first copy that's been identified of that gene, the pseudogene. What I'm showing you there is the distribution of that pseudogene in different species of primates. Humans, chimps, and gorillas, you can recognize that pseudogene in the genome. It's easy to see. Orangutans, monkey, and rhesus, and capuchin monkeys, and hamsters, you cannot find that pseudogene. So it's either degraded or the pseudogenization of that gene occurred in the common ancestor of humans, chimps, and gorillas. Okay? And you can see that these are, look at the pattern of pseudogenes in these different species. This is a pseudogene that's found in all of them, all the species I showed you. So the idea being that the pseudogenization of that gene occurred in the common ancestor of all the species, including the hamster. But the point is that the pattern of observation in these pseudogenes matches the phylogeny, so what we think is the relationships of those species. Right? So the idea here is that each pseudogene is homologous. It's homologous in the sense that this bone is homologous across many vertebrates. Okay? And here's another example of a homologous trait that you can find in all of life. Right? So are there any characteristics that you can find in any living critter? And the answer is there are some. And one is the nature of the genetic code. Okay? So they call it the universal genetic code because I mean, there's always exceptions to these things, but it's almost universal. Right? There are actually variants of the genetic code. Your mitochondrian, the DNA in your mitochondria actually use a variant. There's some small differences in the genetic code that your mitochondria use. You actually have two genetic codes that are used in a cell. But the point is that this would be an example of a characteristic, a homologous trait that you can find in bacteria, E. coli, and humans, or any other species. That's a pretty profound statement, I think. Another thing that Darwin was enthused about is that his theory could explain similarities in how organisms develop. I can't move this up, I'm sorry. In how organisms develop. Right? The observation being that species, closely related species often go through very similar stages in development. Okay? And here's an example of a pattern of similarity in development that was discovered in the 1980s. It's pretty profound, I think. And it was basically developmental biology. It was a lot of developmental biologists on campus, very good ones in fact, who are interested in how you get a very complex adult creature from a single fertilized egg. Now, if you think about that, that's a pretty remarkable process, and that process is called development. And there's a lot of things that occur during development, which if you were to take a course on development, you'd learn about. But one interesting thing is that there's a period of, like in a Drosophila, in a fruit fly, there's a period where you have developing larvae, you have genes that are expressed that set up segments. They're called parasegments. Okay, so you have a segmented larvae, if you will. And then there's genes that are expressed differentially in these different parasegments. And these are called homeotic genes. And the idea is that these homeotic genes act as switches for other genes downstream. So certain combinations of expression of these homeotic genes tell you that you're a head, or you're the tail segment, or you're the first thoracic segment. And then those activate other genes that would turn on the development of antenna or wings or whatever's appropriate for that segment. It's actually a really cool process. Right? So that's what they knew about the development in Drosophila. They actually had identified these homeotic genes. They all have different names. So I put down some of these homeotic genes that were discovered. You can see on the top we have an illustration of the parasegments, the segments, and this is showing you where these genes are expressed. Okay? And the interesting thing is, in genetics, you always discover how a gene works by what goes wrong when the gene doesn't work. Right? That's basically it. It's kind of like geneticists sort of work in the following way. If you wanted to learn how cars work, I mean, one way of learning is just to have a master mechanic tell you how cars work. Right? But imagine you had a lot full of identical cars, maybe hundreds of thousands of them, right? And you're trying to figure out what a certain part of the car did. Well, one thing you could do is you could learn how to turn the car on and off. So, okay, it's working. And then you go in with a hammer and you whack the hell out of that part of the car. And then you see if it works, right? And if it doesn't work, then you can try to figure out what exactly it is that's went wrong. So I think geneticists are a little bit more sophisticated than that. But what they do essentially is they try to figure out what a particular gene does by what happens when something's wrong with the gene. So what goes wrong when these homeotic genes are misexpressed? Well, what you get is you get legs growing where antennae should be. They're really cool mutations, right? So here's an example of I think it's called the bithorax mutation. It's transforming the third thoracic segment of the fly to look like the second. So flies, as an aside, only have one pair of wings. Here's a fly with two pair of wings, right? So you get crazy mutations like that. Here's another one I just told you where the poor fly over here has legs coming out where his antenna should be. Kind of sucks for the fly. But that's an example of that's what happens when these segments are when these homeotic genes are misexpressed or something goes wrong with them in Drosophila. Well, the remarkable thing is that you were segmented at one time too during your development. Right? They don't call parasegments. I think they're called seminomeres in vertebrates. And it turns out that we have homeotic genes. They don't call them homeotic genes because they're independently discovered. They're called Hox genes, but they're the same genes. They're homologous genes in flies and humans. They're expressed in the same sort of way where you have some Hox genes expressed in the head region and so forth. And it appears that they're sort of these master switches just as they are in flies. And interestingly, when something goes wrong with a Hox gene, the effect is similar. So it transforms, for instance, a segment that's further towards the tail to look like a segment that's further towards the head. Right? And so, for instance, here's the H1H box mutant. It makes the hindbrain look like the forebrain in this, I forget what this is, Xenophis, I think. All right. So they could explain biogeography, homology, developmental patterns. And these are not trivial patterns. Right? These are pretty profound similarities between vertebrates and insects. And if you look at an insect and a vertebrate, I can find them to have eyes. Right? There's very few things that look the same, but it's remarkable to think that they actually have some very they're conserved in some of their aspects of development. They share the same, some aspects of their development are the same. I think that's pretty profound. And of course, Darwin had no clue about that stuff, right? But based on other types of similarities in development, he was able to explain that. So I'm giving you the more interesting modern observations rather than the old-fashioned ones that Darwin had. And vestigial organs. All right, so these are organs of an organism that appear to serve no purpose but are there. And as an example, if you give me the slides, this is a pretty cool one. Here's whales. And if you look at a good preparation in a museum of a whale skeleton, that is to say, they articulated all the bones, they made some sort of frame to keep them all in place, and usually they're hung from the ceiling because they're so big. But if it's done well, you'll see usually from 2 wires suspended like a little handful of bones. Right? That's the vestigial pelvis of a whale. So why do you have these little tiny bones that appear to serve no purpose in a whale? Well, the idea is that the ancestors of whales had 4 limbs, just like we do. They're tetrapods that just happen to go back and adopt an aquatic lifestyle. And as an aside, I'll talk about this later as well, but as an aside, whales for a long time were really problematic, not because we didn't think that, that there's something weird about whales in terms of that evolutionary theory couldn't explain, but rather there's a lot of people that are interested in how species are related one to another. If you look at whales, they are so extreme in their characteristics that it's very difficult to find any characteristic that whales share with some other presumably close related mammal species. It's just because they're so weird. And so that's to say, if you look at the characteristics, what characteristics of the forelimb do whales share with their closest relative? I have no idea, but it's very difficult to find any. Just because they've been so extremely modified through time by natural selection. And there's certainly nothing in the hind limbs that you can look at that in whales that will allow you to link them to other mammals. Okay? So that's how the field stood for a very long time. Molecular sequence data came around, and one of the very first things people did is said, let's sequence some DNA, some genes from whales, and use the genes to place them on the evolutionary tree of life. And when people did that, they found out that cows and whales are very closely related. Whales are kind of like swimming cows, if you will. But cows belong to a group of mammals called artiodactyls. Okay? And there's a couple of characteristics of artiodactyls that are out there, but one very key characteristic is they have a very specific ankle joint. Right? They have what's called a double polleus astragalus. It allows them to be really springy animals. Okay? They have this very characteristic ankle joint that only artiodactyls have. It's a characteristic that unites all artiodactyls. Well, whales don't have that double polleus astragalus, at least modern whales don't. And that's where it's good. People say, well, we think they're closely related to cows, and it's only based on these DNA sequences. Well, in the last 10 years, that story has changed remarkably because there have been a lot of discoveries of whale fossils that go back up to 40 million years ago in places like Pakistan, easy places to get to, and Egypt. Okay? And these whales actually have hind limbs, and those hind limbs have a double polleus astragalus, which is pretty cool. All right. What is the time? I spent so much more time on that than I thought. I'm going to start population genetics. Blank the screen up, turn on the little lights. I want to talk briefly about, so that is basically the origin of species. I mean, I recommend it's not a bad read. It's very Victorian in its writing, but it's not difficult to read. There's nothing mathematical in the book. There's nothing, it's an accessible book, and there's a lot of fields where if you look at the founding works of those fields, they're almost impenetrable. Right? So if you look at some of the founding works in physics, you have to be a pretty good mathematician to really penetrate that work. Right? That is not the case with the origin of species. Anybody can assess the book. Anybody can understand it. The arguments, as I've outlined in here, are pretty straightforward. Okay? So the first thing I want to talk about was what was the initial reception of Darwin's theory by other scientists? And remembering that there's different parts of a successful theory of evolution: the fact, the pattern, and the mechanism. And people, well, almost everybody immediately accepted the fact of evolution. Right? Like I said, evolution was not a new idea to Darwin, but he amassed so much additional evidence that people really were sort of confronted with it and had to, they accepted it for the most part. Okay? But there was debate for a long time about the pattern and the mechanism. So it's remarkable because I told you that the main contribution Darwin made was this mechanism, natural selection. Well, that was not immediately accepted by his peers. Right? This idea that you have variation which is generated randomly being filtered by this non-random process was still, a lot of people have problems with it today. They shouldn't, but they do. That was a problem for some people back then too. Okay? And remembering too that Darwin put a lot of his money down on the pattern of evolution being slow and gradual. That is to say, if you have a trait in time, and you have two, a starting point and an end point, Darwin made this argument that the change was slow and gradual. And going back to the problems with transitional forms, his argument would be that there would be, or had to have been these intermediate states, and each of these intermediate states had to be better than the previous state. Right? I'm just repeating what I told you earlier. But there are some types of characteristics, like I said, as an example with wings, where there's sort of a critical threshold. Either, as an example, your wings are long enough and you fly, or they're not long enough and you fall to the ground. Right? You don't fly. So there are some characteristics like this where it's more difficult to explain those types of changes with slow and gradual change. And a lot of his peers point out the following, which is, what, if you would just give up on this, Charlie, if you just give up on this idea of slow and gradual change, this problem would go away. If you have a pattern like this, well, then in one fell swoop, you might be able to go from no wings to wings and flying. And the problem with that is we don't think this is how things really work in terms of complicated structures evolving all at once. No modern biologist thinks that. But this is the type of argument people were making, and there was even this idea of remembering this is a period where people don't even understand genetics, much less develop it, right? So the idea being, well, why not have these big changes? Maybe that's a biological process that really occurs. They didn't know any better, but that's an explanation that could fly at the time. So there was this debate about the pattern as well, okay, immediately after the origin of species. And then there was this other problem with this theory that people pointed out, which was heritability. Right? So Darwin's argument relies on the fact that traits are heritable. Right? And he relied on this idea that was just common knowledge. Big dogs give rise to have pups that are big. Right? That was the argument. But that's not a very sophisticated argument because it's very natural to say, well, why do offspring resemble their parents? Why is that the case? And Darwin, like everybody else at the time, had to say, I don't know. They just do. That's not very satisfying. So it turns out later, in later editions of the Origin of Species, and went through many editions over his lifetime, he kept revising the text of the book, and he came up with some pet theories about how heritability really worked. And it turns out these theories were wrong, dead wrong. So his ideas they developed later in life on how why offspring resemble their parents were wrong. Right? And in fact, they actually, from a theoretical point of view, they actually contradicted his theory because he had this idea that here's our trait again. He had this idea that inheritance was blending. That is to say, it's kind of like mixing colors of paint. So you have red paint and you have white paint, you mix them together, you get pink paint. Well, you mix pink paint with pink paint, you get more pink paint, right? You don't get red or white, you can't get the extremes back out. So his mechanism, if you were to make a pink flower or a red flower with a white flower as an example, you would get a pink flower. And so the next generation, you'd have much less variation just from random mating. Under the system of inheritance, you would keep reducing the amount of variation in the population. Remember, natural selection is a process that depends on there being variation among individuals to actually work. If there's no variation from one individual to another, there's nothing for natural selection to grab onto and change, right? So his ideas that he developed later were based on blended inheritance. They were flat wrong, and people later on pointed out that they even contradicted mathematically his theory. Okay? That's not to say his theory is wrong, but just his ideas about inheritance are wrong. And in fact, if you really want to read the best version of the Origin of Species, read the first edition, which was unsullied by these incorrect ideas. So interestingly, the answer for how why you have offspring resembling their parents and how heritability actually works was in his study, right? Because in his study, he had a paper by an Austrian monk by the name of Gregor Mendel. And you guys have all heard of Mendel because you've all taken biology in high school. In 1865, Mendel published his results of his experiments on breeding of peas. Peas, because they're easy to keep in a garden, you can manipulate which male, mated with which female pea plant. All right? And so the answer was in his study. It turns out it's not, there's two possibilities. The incorrect explanation that I heard a long time ago was that he just never read the text. It's not clear that's true or not. I actually talked to an expert on Darwin, this woman who's now at Harvard, but was the person who was the curator at Down House, which is where Darwin lived later in his life. And it's probably the case that if he read Mendel's works, he just didn't appreciate what it meant. Sometimes people read things and they just don't get it, right? And evidently he must have read, if he did read this work that he had in the study, he just didn't get it. But the answer is right there, right? And Mendel's ideas are that instead of having blending inheritance, that you might call it particulate. That is, there's bits that are transmitted from mom and dad, right? So Mendel was the guy who introduced the idea of a locus. That is the site that has the code for a particular trait. Today we might call that a gene. But the idea in a modern sense, a gene can be much more than, a locus can be much more than a gene. For instance, you can have a locus that is just one nucleotide position in your genome. So remember, your genome is about 3 billion ACGs and Ts in length. You can actually say my locus is going to be the nucleotide at position 1,452,563,444. That would be a perfectly valid locus. And then the variants at that position could be the different ACGs and Ts you might see in a group of individuals in a room, say. And those actually have a name. They're called SNPs. But I'm going to continue this discussion this Friday.